All right, let's dive right into this explainer. Because today, we're talking about something that honestly feels straight out of a massive Hollywood blockbuster. But it's actually unfolding right now in high-tech labs all around the world. I'm talking about de-extinction. Now, for centuries, the word extinct meant forever, right? It was the ultimate biological finality. Case closed. But thanks to some absolutely unbelievable leaps in genetic engineering, that absolute finality, well, it's being challenged. We are currently standing on the precipice of what scientists are calling the Lazarus Surge, this period where species we thought were gone forever might literally walk the Earth once again. It's fascinating, it's highly complex, and yeah, as you might imagine, it is incredibly controversial. So, here's the roadmap for our explainer today. We're going to hit the 2026 timeline for this Lazarus Surge, meet the actual de-extinction candidates, break down how CRISPR makes this whole thing possible, look at the fierce science versus nature debate, and finally wrap up with the ultimate moral question. Okay, first up, the 2026 Lazarus Surge. To really grasp the sheer scale of time we're playing with here, just think about the historical gaps we're trying to bridge. I mean, we are connecting the deep past with the immediate future. The woolly mammoth vanished roughly around 2000 BCE. Just to put that into perspective, when the last mammoth died on some remote Arctic island, the Great Pyramids of Giza had already been standing for centuries. Then, jumping forward, we have the famous dodo bird, totally lost by 1681. The South African bluebuck wiped out around 1800, and the thylacine or Tasmanian tiger, whose last known lonely member died in a zoo in 1936. For decades, or millennia in the mammoth's case, those dates marked a tragic, definitive end. But check out that final milestone, 2026 CE, the target date for the Lazarus Surge. This is the exact year certain biotech companies are confidently aiming to introduce their first resurrected proxy species. We are contrasting thousands of years of absence with a world-changing target date that is literally right around the corner. Now, before we get to the specific animals, let's make sure we're totally clear on our terms, because the reality here is a bit different from the movies. De-extinction is defined as the process of creating an organism which is either a member of or closely resembles an extinct species. Notice that extremely careful phrasing there, closely resembles. We aren't finding a perfectly preserved mammoth frozen in a block of ice and, just waking it up with a warm blanket. Actually, we're talking about creating a proxy. This is a highly engineered, modern organism designed from the ground up to fulfill the exact biological and ecological role of its extinct predecessor. The goal isn't a theme park attraction, it's targeted ecological engineering. We're essentially hacking a modern animal so it can survive in a lost habitat, trying to repair ecosystems that fell totally out of balance when these majestic creatures disappeared. So, let's meet those de-extinction candidates. What's really mind-bending here is how this biology actually works in practice. To bring an animal back, you can't just conjure it out of thin air, right? You need a living blueprint, a genetic anchor. Basically, scientists have to pair these extinct marvels with their closest living surrogate relatives. So, to get a woolly mammoth, they are working intimately with the genome of the Asian elephant. But wait, look at the next one. For the carnivorous thylacine, they're using a tiny, mouse-sized marsupial called the fat-tailed dunnart. Think about how wild that is for a second. A tiny marsupial potentially serving as the biological foundation for an apex predator. The iconic flightless dodo is paired with the beautiful Nicobar pigeon. And the newest addition to the de-extinction list, the South African bluebuck, is being paired with the roan and sable antelope. Without these living relatives acting as genetic guides and potential surrogate mothers, this whole endeavor would be completely impossible. To give you a real sense of the massive ambition driving this movement, listen to this declaration from Colossal Biosciences. They're the leading biotech company operating in the space right now. Speaking about the bluebuck, they state, "The plan is definitely to make a living, breathing bluebuck, a biological marvel." The bluebuck is a super fascinating case study. It was hunted to extinction by European settlers around 1800, a direct casualty of human expansion and habitat loss. But Colossal isn't trying to make some fragile museum exhibit that lives in a sterile bubble. No, they want to reverse that historical biodiversity loss. Their vision is to engineer these animals to be exceptionally healthy, resilient to modern diseases and climate shifts, and fully capable of surviving in the wild. The ultimate finish line? Rewilding. Reintroducing massive, thriving herds back into their historical grazing habitats to naturally manage the landscape once again. Which brings us to the big question. How exactly does CRISPR resurrect species? Okay,
The absolutely crucial piece of this entire puzzle is CRISPR. It's defined as a powerful tool for editing genomes, allowing researchers to easily alter DNA sequences and modify gene function. Let's just pause to appreciate this for a second. Before CRISPR, this entire concept of de-extinction was literally impossible, a total dead end. Think of an animal's DNA like a massive, incredibly complex encyclopedia of instructions for building a living creature. For the longest time in human history, we could read that encyclopedia, but we absolutely could not edit it. CRISPR changes everything. It acts like a highly precise, microscopic word processor. It lets scientists find one specific sentence in the DNA, safely cut it out, and paste a brand new one in. It's the revolutionary key turning this pipe dream directly into reality.
So, how does this actually play out? It builds across 3 monumental steps. It sounds simple, but the science is just staggering. Step 1. Sequence extinct DNA. Scientists extract whatever fragmented, degraded DNA they can scrape together from museum bones or ancient frozen remains, and they painstakingly map it out. Step 2. Edit the surrogate genome. Using that ancient map from step 1, they take our molecular word processor, CRISPR, and heavily edit the DNA of the living relative, like the Asian elephant. They artificially tweak the elephant's genes to express mammoth traits, like a thick, shaggy coat, smaller ears to prevent frostbite, and extra fat layers for extreme cold. Step 3, implant in a surrogate mother. Once the DNA is edited, an embryo is created and implanted into the living surrogate to carry to term. And guys, this is a massive biological mountain to climb. An Asian elephant has a 22-month gestation period. Managing a genetically modified pregnancy and a massive, highly intelligent, endangered animal for nearly 2 years is a logistical triumph all on its own. But if all goes well, an animal resembling the extinct species takes its first breath.
Now, let's look at the science versus nature debate.
Because anytime we gain the power to alter biology on this unprecedented scale, incredibly serious ethical questions arise. We find ourselves looking at 2 deeply divided perspectives here. On the pro-de-extinction side, advocates passionately argue that bringing these animals back restores lost ecosystems. Mammoths, for example, could help trample snow and knock down trees to maintain the Arctic tundra, which is absolutely crucial because that permafrost traps massive amounts of global carbon. Proponents also argue it's a moral imperative, a way to correct historical human mistakes, since our hunting directly wiped out creatures like the thylacine and the dodo. But on the anti-de-extinction side, critics point out a glaring issue. This technology drains crucial conservation resources. Creating just a single engineered proxy costs millions upon millions of dollars. Critics argue that money could and should be used right now to fund anti-poaching units and protect the critically endangered rhinos and elephants that are clinging to survival today. Plus, they argue it dangerously meddles with natural selection, risking the introduction of engineered species into fragile modern ecosystems that have already moved on and adapted to their absence. And this counter perspective is brilliantly put into focus by organizations like the Wilderness Society. They state, "We must focus on protecting existing habitats and natural selection, rather than diverting resources to resurrect the past." It's an incredibly powerful argument that demands our attention, doesn't it? It asks us to seriously consider whether our technological fascination with correcting the past is blinding us to the urgent, desperate conservation needs of the present. Are we essentially playing God in a pristine, high-tech laboratory while the real, natural world burns right outside the window?
Which leads us perfectly to our final section, the ultimate moral question.
All of this science, history, and fierce debate forces us to confront this. We've traced the timeline, met the animals, marveled at the CRISPR revolution, and weighed the profound ethical costs. And it all boils down to this. We finally have the power to reverse extinction. But just because we can, does it mean we should? By 2026, the Lazarus Surge could present us with a living, breathing biological marvel stepping right out of the shadows of the past. It will undeniably be a triumph of human ingenuity, for sure, but it will also be an unprecedented test of our collective wisdom. As we step over the threshold into this astonishing new era of genetic engineering, we have to decide exactly what kind of stewards of the planet we want to be. I'm going to leave that question with you to ponder. Thanks so much for joining me on this explainer, and keep questioning the incredible world around you.